Stephen Durand, August 10th, 2021. So I've decided to do my presentation on sperm maturation in the germinal epithelium and its hormonal control. So first, a little bit about the germinal epithelium. It's the epithelium in the seminiferous tubules where sperm is produced. Sperm cells arise from the division of spermatogonia, which are basically the sperm stem cells that divide. The germinal epithelium also contains Sertoli cells that aid in sperm maturation by providing sperm with the right environment and growth factors to divide. So in this diagram, this would be the lumen of the seminiferous tubules, and this is the germinal epithelium. And so at the basal layer, you have the spermatogonia that divide to form the sperm cells. And so as the cells divide, they're pushed through the epithelium towards the lumina, which eventually the sperm are released into the lumen. So sperm development begins with the division of spermatogonia at the basal layer, and one of the daughter cells of this division stays at the basal layer, and one of them is pushed outwards towards the lumen. So the spermatogonia at the basal layer continues to divide to form two more spermatogonia, whereas the spermatogonia that get forced out, they undergo more mitotic divisions to form primary spermatocytes. And so these primary spermatocytes undergo meiosis, not mitosis, which involves genetic recombination to form secondary spermatocytes. So primary spermatocytes undergo meiosis, one round of meiosis, to form secondary spermatocytes. And the secondary spermatocytes undergo meiosis again, only without DNA replication. So they form, instead of diploid cells, they form haploid cells with half of the original number of chromosomes and amount of DNA. So the result is you have four spermatids. They're no longer spermatocytes, they're now spermatids with half the amount of original DNA. And so these spermatids mature into sperm at the lumen. That at this point the divisions are done. It's now a matter of the sperm maturing, creating the right cellular machinery and such to be released into the lumen. So now that we've talked a little bit about the process of sperm maturation, now we're going to talk about the hormones involved. So testosterone is essential for sperm maturation and is secreted by Leydig cells, which are also called interstitial cells, which are in in between the seminiferous tubules. And so testosterone does a couple things. One of them is it develops secondary sexual characteristics. The other one is that it stimulates Sertoli cells to help in the maturation of sperm. So going all the way up to the brain, the hypothalamus releases gonadotropin-releasing hormone, which, when it reaches the pituitary gland, triggers the release of luteinizing hormone, LH, and follicle-stimulating hormone, FSH, from the pituitary. And so they both reach the seminiferous tubules, and so LH, luteinizing hormone, stimulates testosterone production in the Leydig cells. And so FSH, on the other hand, reaches Sertoli cells and increases the ability of the Sertoli cells to facilitate sperm maturation, and the Sertoli cells also secrete hormones and other products. And some of the other products include growth factors, inhibin, androgen binding protein, enzymes, and products that help in sperm maturation. And one of the things that's important about this is, so testosterone is sort of, Sertoli cells is sort of how testosterone is affects sperm maturation, because sperm itself, the germ cells themselves, don't have receptors for testosterone or FSH. So it's the FSH and testosterone affect sperm cell maturation indirectly when they reach the Sertoli cells. So there's also some negative feedback systems involved in hormonal control of sperm maturation. One of them is inhibin. So when the pituitary produces FSH that affects the Sertoli cells, that secretes its products, which aid in sperm development. It also secretes inhibin, which inhibits FSH secretion by the pituitary. So it's sort of negative feedback loop where, you know, if we have too much FSH, then FSH secretion decreases. Another important mechanism involved in FSH is when FSH stimulates Sertoli cells, they release androgen binding proteins, which bind to testosterone, which keeps testosterone in the lumen, because when androgen binding protein binds to testosterone, testosterone can no longer cross the cell membranes and is trapped in the lumen, which is important because testosterone itself is an inhibitor of gonadotropin releasing hormone, which indirectly reduces the activity of Sertoli cells as well as interstitial cells or Leydig cells. And it's a direct inhibitor of luteinizing hormone, which inhibits the activity of interstitial cells or the testosterone production. So that is another negative feedback loop. And these are my reference. I hope you've learned something today.